Thank you so much, everyone, for joining us tonight. We'll be getting started momentarily. Thank you so much for joining us. We'll be getting started in just 1 minute. Hello, everyone. Good evening or good afternoon from wherever you are. Thank you so much for joining us for Ask Me, our educational webinar series. My name is Audra Hines, Vice President of Care and Advocacy for the ALS Network. I want to thank our lead sponsor, Mitsubishi Tanabe Pharma America, as well as our mission benefactor families and everyone who helps support our programming. The contributions from these individuals and sponsors and families are truly amazing, and programs like this one tonight, as well as the multitude of services we provide, would not be possible without these generous donations. May is ALS Awareness Month, and we want to ensure you know that the ALS Network offers a menu of services free of charge to people with ALS and their families. Please know that if you need a piece of equipment loaned to you, emotional support or counseling, assistance with finding home care, help understanding insurance benefits, guidance in finding an ALS specialist for care or a clinical trial, or just someone with expertise to answer any other questions about the disease, the ALS Network is here for you. Our committed staff who are locally based in California and Hawaii provide high quality, personalized and local services as we also fight on the legislative advocacy front while building our growing research program. We're your local team and we're glad to provide this multi-layered support. To learn about the ALS Network and programs and services we provide, please visit alsnetwork.org for more information. We're really pleased tonight to have a special guest speaker. Dr. Christine Vande is full professor in the Department of Neurosciences at the University of Montreal, as well as the scientific director of the Packard Center for ALS Research at Johns Hopkins. Her research interests are centered on understanding the underlying pathological mechanisms that lead to ALS and FTD. We were thrilled to have her here with us last year too in providing this important research update. Thank you so much for being here, Dr. Vande.
It's my pleasure. Thank you so much for having me and for hosting this important information session. I am thrilled to be here and working with you guys on this. I'm going to put my slides up on the screen. And with that, we'll get going. So, so thank you so much. So I'm here to talk to you a little bit about the Robert Packard Center for ALS Research at Johns Hopkins, as well as to tell you about our annual meeting that we hold every year in March, in which we actually host over 300 people in Baltimore, plus another 125, 150 people online from more than 70 different institutions around the world, all coming together, focused on trying to understand the biology that drives ALS, as well as developing therapies and biomarkers. So the Packard Center, just for those that maybe are not familiar, is really an organization that is focused, has a vision really of focusing on altering the course of ALS, giving those diagnosed the opportunity to live long and healthy lives. And we do that through our mission, which is empowering collaborative and breakthrough research that advances our fundamental understanding. And we do that by engaging and supporting the global ALS research community. This year's meeting was held in March in Baltimore, and the prevailing theme for this year, which was our 25th anniversary of Packard, was all structured around hope. And to kick off the meeting and was actually two very, very, very important speakers. So Dr. Richard Bedlack from Duke University, who probably many on the call are familiar with. Dr. Bedlack speaks a lot about hope in his practice and the impact that hope has on patient experience with ALS, as well as Ms. Cassandra Haddad, who works at Temple University and is actually one of the founders of Vande Legacy. These two individuals gave us very, very inspiring talks about what hope means to them, both from someone who meets with patients every day and from somebody who actually is a gene carrier, and what this means to her family over the years. And so the tagline of Packard is "the hope is in the science." And so hence why we really grounded the meeting this year in this particular theme. So here's what I was telling you about the meeting, about how much engagement there is at this meeting. So we bring together global researchers from all over. And so we had 10 different countries represented. We had researchers from Germany, France, the UK, Italy, Israel. I think we had someone from Sweden. I don't remember now all of them. It was a very, very dynamic 3 days of nonstop science. Basically, it's 46 presentations packed into 3 days. It's also accompanied by 2 poster sessions in which young trainees, like postdocs and students, these are people that are training to eventually perhaps one day run their own research lab or contribute to industry or work in the nonprofit sector. They were all there presenting their latest unpublished research, almost all unpublished research on ALS. We did that over 2 evenings. We also had a networking lunch in which these young trainees were able to engage with leaders in our field, and both from academia as well as industry and the nonprofit sector. So really trying to foster this next generation of researchers. Our keynote speaker was this amazing woman, Dr. Beth Stevens she's Harvard. She actually has contributed really important research to many different neurodegenerative disease spaces, including ALS. And she gave a very inspiring talk about some very specialized cells that we all study in ALS called the microglia, which are sort of these cells that serve to stand guard over the neurons in our system. So she gave a fantastic talk and really helped to kick off the meeting. So what is all this research that we're doing, that we're working on? So I just want to put this slide up as a way to sort of get us all on the same page. Packard focuses on fundamental biology. Some people call it basic research, but I always say there's nothing basic about research. It's fundamental. It is the foundations on which all therapies and biomarkers are based. And basically, our idea is that we need to figure out how this very, very healthy neuron got to be so very, very sick and dysfunctional. And if we can understand what went wrong here, then we can understand how to stop that and make these sick neurons maybe perhaps better again. That's basically what we're looking for. Is we're looking for true disease-modifying therapies that return these unhealthy neurons to a happier, healthier state where they're firing properly and communicating to the muscles and much better than they were. So this is the primary focus of a lot of the research that's associated with the Packard Center. So the Packard Center basically feeds a pipeline. And so some of you are familiar with this idea of this progression, probably more here, and from talks you may have heard from others about clinical trials. There's phase 1, phase 2, phase 3, and then there's a review by the Food and Drug Administration, the FDA, to approve drugs. But way before a drug ever gets into a patient, there's a whole bunch of other research that happens first. And that it starts with the identification of a target. So what is that biological target that might be different, that could be potentially targeted for a therapy? And then there's going to be an assay or tests, experimental tests that are designed to try to understand how do you interrogate the function of that target and determine and identify molecules that can improve the function of that target. And then
you're cutting out just a little bit.
It drives this early. Oh, okay. I'm sorry. Am I okay now?
I can hear you, but you're frozen a bit.
Yes.
Yeah.
Is it okay now, Audra?
I can hear you okay. I can see your slide. You're frozen, but hi there. I can see you, but I can't hear you.
Can you hear me now?
Yep, perfect.
Yes? Okay. We switched to a different source of internet.
Okay, terrific. Thank you so much.
Yep. Apologies for that. Okay, so going back to how much of this part was understood?
You might want to start over on this.
Okay.
Yeah, that's great. Thanks.
Okay. All right. So this slide was meant to really sort of point out sort of how drug development happens. And so many of you on the call probably are familiar with and have heard a lot about phase 1 or phase 2, phase 3 clinical trials and the idea of drug approval through the Food and Drug Administration, the FDA. But long before a drug ever reaches a patient, there's all of this other research that has to happen first. So, for example, when I pointed out about that healthy neuron, and as it becomes sick, and we're looking for what are the mechanisms that are driving that neuron to become unhealthy, that's part of this target identification, is trying to understand what is the pathway that exists. And then the next part of that in the pipeline, if you will, is trying to identify molecules that might help. And so this is what they call screening. And so you make a particular type of experimental assay that you could then determine whether or not the molecules are being effective at rescuing whatever function might be compromised. And then this third component here is basically refining that second step in something called a hit to lead program. This part is usually undertaken by industry, and Packard is usually right here, sitting in the very front end of this pipeline. Packard really drives early stage research that feeds this therapeutic development pipeline. And in fact, many of the therapeutics that are moving towards trials these days have come from research that's been done in academic labs. Most of this is done in academic labs, and Packard is very privileged to be able to support several of these works in their very, very early stages. Sometimes actually providing, that seed funding, that catalyst funding that actually generated the early data that allowed that work to really develop and be supported by larger sources. So Packard sits right here in the pipeline. This is what most of the Packard meeting is about, is trying to figure out this part of the pipeline. So how do we do that as a field? We do that by studying genes, actually, a lot of the time. So as I'm sure most people are aware on the call, there is a certain proportion of ALS is familially inherited. We call there are particular genes that have variants in them that lead to ALS. We also understand now that there are some of these variants also contribute to ALS in instances where there's not an obvious family history. We understand that when we get these genes, we try to put them in buckets of different mechanisms that they might be interacting with. And you'll see that for some of them, they actually fall into more than one bucket. And that's sometimes these things do many more than one thing. My point here that I wanted to show, and this is not up to date because there are many more actually puzzle pieces that have been filled in the last few years since this figure was first published. But I wanted to point out is that sort of the big four genes that are studied in the ALS context are these ones: the SOD1, TAR-DPP, which encodes TDP43, FUS, and then of course C9orf72. There's a significant amount of research in the field that is invested in that. The other genes are also investigated, and they're also represented in our Packard meetings. We try as much as possible to make sure that there's lots of diversity, because it's only by understanding the mechanisms from many different angles that we're going to get to some common understanding of what has gone wrong in these neurons. So understanding ALS biology, I'm going to show you something that I don't want you to be freaked out, but it's not straightforward. So this is a beautiful analogy that I've borrowed from a colleague here, David Taylor from ALS Canada, who talks about how if we think about understanding ALS biology, think about a jigsaw puzzle. And in 1993, this is when the SOD1 gene was identified, is when we just kind of got the box, got the puzzle picked out. And then you see in 1993 that we discovered SOD1, and so you start to see like, oh, I got the puzzle pieces out of the box. And then 2006 was the major discovery about TDP43 contributing to the disease. And then shortly thereafter, there was FUS and C9orf72. And then a broader understanding of what these things do. And this is where we are today. So you see that we don't have the full picture yet, but there's been steady progress of filling in the various pieces to understand what is the underlying biology. And this is done through a series of different types of approaches where groups use cells that are cultured on plastic dishes. Sometimes those cells actually originated from patients, and they've been what we call pre-programmed so that they basically look like some of the neurons that are lost in ALS. We do that by using mouse models. We do that by using fly models. Zebrafish, which is this cute little tropical fish that is very popular in several groups. We also look at, the most important tissue of all, which is the tissue that comes from patients. And so all of this helps to fill in these puzzle pieces. And that is actually, I would say 95% of the talks at a Packard meeting are using these models. So all these different puzzle pieces, how do they fit in? So they lead into these various mechanisms, which I alluded to in that first puzzle image. Here now it's kind of trying to superimpose some of that onto the motor neurons, which are the neurons that are lost in ALS, but as well as, there are these other cells that are really important to supporting these neurons. So there's astrocytes or microglia. Of course, there's the muscle itself. And all I want you to take away from this is that there's a lot going on. There are lots of different genes that have been implicated, which means there are lots of different mechanisms implicated. Some things can have a role in more than one mechanism. It's complex. It's not straightforward, but I do want to reassure you that there is steady progress that is being made as we start to fill in more and more of this particular picture. So to the meeting. So highlights from the meeting. And so I'm going to walk you through sort of three main sections of the meeting. The first one, and this is going to look very similar to my last year's talk, is the focus on TDP-43 biology. And it just has to be a separate section because there's just so much of the field that is dedicated to understanding TDP-43. And we'll go over what that is in a minute. I'm also going to tell you about some newer technologies that were utilized in various projects, and a couple of stories that I think are sort of new and exciting. So I'm going to tell you about the investigator, a little bit about their projects, and I'll be happy to answer questions at the end. So TDP-43, what is it? What is cryptic splicing? And so it's kind of like we're getting these hieroglyphs, right? In this cartoon, it's these secret messages that exist in our systems, and trying to understand what that means. So TDP-43. So TDP-43 is it's called an RNA binding protein, and we'll talk about what RNA is in a minute. So it's normally found in the nucleus of a neuron, neurons as well as many other different cell types. But then the nucleus is the command center of the neuron. It is where all the genetic material is stored. In fact, TDP-43 helps to keep that genetic material in good shape. And so in brown here, this is an actual section of a spinal cord, piece of spinal cord from a postmortem material. And we see there it is in the nucleus. It's this brown labeling. And then you see that there's a little bit sitting out there in this other region, which is called the cytoplasm, but it's not a lot. It's mostly there. And that's what it does, is it sits there and it governs how that genetic material gets utilized. I always call it, it's the master regulator. And the way I see it is that it's because if this particular protein is not there, lots of things go wrong. And so in the context of ALS, as well as frontotemporal dementia, and now as we understand other neurodegenerative diseases as well, TDP-43 is not in the nucleus where it should be. So here, for example, if you look at this neuron, this big empty spot here in the middle, that's that nucleus command center. And you see now the brown labeling is everywhere else but there. You see that here as well, here. You'll also notice that there can be different types of patterns. Sometimes they're small. They're these round puncta. Sometimes they're sort of granular. There's big things and there's fibrous things. There's lots of interesting work that is correlating these different structural patterns to the different mechanisms that might be implicated. Many groups are doing that now. Very, very exciting. But this is sort of the key pathological signature. So when a scientist looks at a piece of tissue that they've labeled for TDP-43, this is one of the key findings that is going to be found in the brains or spinal cords of patients that have ALS. And in fact, so much so that it's 97% of cases are going to have this particular feature. And so this is why there's such a strong emphasis on trying to understand what is the fundamental function of TDP-43. So here, as I mentioned, so 97% of the ALS cases have this particular mislocalization of TDP-43. And actually also of note is that frontotemporal dementia, FTD, there's also about 50% of the cases will also feature this mislocalization of TDP-43. So really, really fundamentally important. This has set up a big debate in the field about whether or not this movement of TDP-43 out of the nucleus into the cytoplasm constitutes a loss of its endogenous function, so a loss of what it's supposed to normally do, or did it gain some sort of toxicity by going to this other compartment? And there's still a lot of debate about that in the field, although I'd say that folks are now starting to gravitate hard towards the idea that it's the loss of its nuclear function, which I personally also subscribe to as well. So what is RNA and why is this so important? And so this is an analogy that I've used in the past, in which if we think about the nucleus as the command center, that's where everything is. This is the big recipe book. This is all the family heirloom recipes are sitting right there in that nucleus. And what you want to get to is the end is this beautiful cake that grandma used to make. But to get there, you need to go through an intermediate step, which is you need to find the recipe in the first place. And so that recipe card is that intermediate piece. That's the RNA. And so the DNA gets translated or transcribed rather into RNA. And so it's decoding it. And so that the cell can then use this intermediate recipe to generate this beautiful cake or the protein. And the proteins in our cells are what do most of the work and keep our neurons happy and functioning and healthy. So TDP-43 is central to understand to regulating these particular intermediates. All right. Okay. So now let's get to some of the very cool stories in which TDP-43 has been shown to have a role in how it regulates those intermediate RNAs. And so cryptic splicing is the idea that if we have pieces of information that in our RNA has to be stitched together. And so basically, you got to get rid of sort of the junk sequence in the middle. And TDP-43 actually um, is important because it actually prevents the inclusion of pieces of the RNA that normally would be thrown away. And so these are so-called cryptic exons. And so in this particular case, UNC13A is a new target. It was discovered about a year and a half ago now by this guy, Pietro Fratta. He's from University College London in the UK. And this UNC13A protein is super important for helping neurons to fire so they get the neurotransmitter to the cell surface so that they can communicate to the next neuron. When TDP-43 leaves the nucleus, this junk piece of RNA now gets included. And when it does that, it actually results in a loss of the final protein product. And so we don't get the proper neurotransmission that is supposed to happen. So new this year was that, Pietro, Dr. Fratta, he reported that he's identified antisense oligonucleotides that can modulate the splicing and return it to this normal state, and actually showed that it can then rescue this deficit in neurotransmission, which is fantastic. And in fact, he founded a company called Trace Neurosciences that has just raised $100 million to initiate a phase 1/2 trial. So stay tuned because I think we're going to be hearing more about that probably in the next year. Dr. Fratta also showed some very new and exciting data about other types of cryptic splicing events and other genes that generates new signals that can be recognized by the body's immune system. It's very early days, but very interesting and exciting work because it really starts to kind of make us question about what is the purpose of these, the inclusion of these so-called junk pieces of RNA and what other functions can there be happening in the body. So very, very cool and exciting new work coming from Dr. Fratta. The next story I'll tell you about is from Dr. Len Petruccelli at the Mayo Clinic in Florida, is the idea, again, based around these cryptic exons. This is in a different target called HTGFL2. I know it sounds like alphabet soup. I just want you to appreciate that what's important to appreciate here is that it's thought that this particular target, rather than it being a therapeutic target, it actually may be a potential biomarker. And so this is a finding. This particular cryptic exon has been identified by two other investigators as well, Dr. Michael Ward at the NIH and Dr. Phil Wong at Johns Hopkins. They both identified this as well. And that's actually really reassuring, is when you see the same type of target that is being picked up by multiple labs using different approaches and different models. It's very exciting. And so it's even being proposed that perhaps that this cryptic HDGFL2 protein that gets generated occurs prior to the rise in neurofilament signal, which is now becoming an important biomarker in clinical trials. So this could be very interesting and maybe help predict, for example, when somebody who's a gene carrier for an ALS gene might convert. Because that signal will rise before the neurofilament rises, right? So when the neurofilament rises, it indicates that there's neuronal damage. And so it'd be really cool if this works out, that it's something that is happening really early and we could detect it. What's new this year is that Dr. Len Petruccelli actually has developed new tools that are 10 times more sensitive than the original ones that were published just 2 years ago. And so the higher the sensitivity, the better that this is probably going to work. And I just put this little cartoon here in the corner just to highlight to you, there are lots of different cryptic exon events that are being followed in the field. And so I told you about the UNC13A, but I'm going to tell you now about this one here, KCNQ2 on the next slide. So another target is this KCNQ2. This is from Dr. Evangelos Kiskinis at Northwestern University. What's different about this one is that it might actually inform on why we think that the neurons are firing too much, something called hyperexcitability. So the idea that the neurons are communicating too much. And when they do that and when they're firing too much, well, they get tired. They exhaust their resources too quickly, And then they kind of get sick. They're like, I'm tired, I need a break. And then what happens is that the neurons actually they degrade eventually. In this case, when there is that inclusion of the junk RNA, what happens is you get something called a dominant negative version. And so basically you get a version of this protein that gets generated that interferes with the way the wild type version of it is supposed to function. So basically, again, you're interrupting neurotransmission from working properly. And Dr. Kiskinis is also developing an antisense oligonucleotide approach to explore as a potential therapeutic. And again, very early. If you remember in that pipeline about target identification, we are at step 1 in this project as they look to see whether or not now they can develop a screen and identify a molecule that could actually improve the firing of these neurons. Okay, so on to the next step on new technology. So cool new tools that we're going to advance our understanding in ALS and make us go faster. And so this is another research project that I have shared with this through this platform before. This is from Dr. Claire Clellan, a young clinician scientist from UCSF in San Francisco. She has just generated a lot of excitement in the field. She's looking to harness a new technology called CRISPR, or CRISPR-Cas9. And basically, without getting into the details, this is a technology that is basically like a piece of scissors that can go in and precisely edit the genetic material in a cell. And so the idea here is that she's focused on the C9 expansion in ALS patients and really trying to figure out how to cut out the expansion. And in fact, so here are some images from one of her papers. And so if you focus here on here, we see that with this tool that she's got, it's called REX. She actually can decrease the number of these little green dots that actually are representative of some of the neurons that have a negative phenotype, that they're not well. And so she has figured out a way in which she can actually restore edit the C9 expansion. And so she can actually put TDP-43 back in the nucleus. And that means now that it can do what it's supposed to do with repressing the inclusion of those junk pieces of RNA, like I showed you in the last section. The update here to this project, there was incredible excitement about this from many people in the room this year, that she's got the mechanism working in cells that have been cultured, so on human neurons that have been cultured on a plastic dish. That's working really, really well. And now she's looking at how can she move this into an intact nervous system? And so she's working with many different groups, trying many different delivery mechanisms using adeno-associated virus or viral-like particles or nanoparticles, many different types of strategies to try to get this to be efficiently delivered into the neurons that need it. And of particular interest is that she's established a human platform to test this new approach. And this sounds a little weird because how would you test it in humans? Because that's what I thought when I first heard it. But Dr. Clellan she is a practicing clinician who noted in her particular clinic that there are often individuals that are cognitively inactive, so brain dead. And so they present an opportunity perhaps to test these tools in a real live human central nervous system. Of course, all of this has been rigorously reviewed by the various ethics committees that are involved. It's in the early days, but she's establishing this, and I think this is a very exciting and innovative opportunity here. So we will definitely stay tuned because this work is definitely evolving just beautifully. Tons of excitement here. Another area of particular excitement was actually another researcher, Dr. Peter Tessier from the University of Michigan. He is actually a new recruit to the Packard Investigator Network, somebody who comes from a much more chemistry, structural biology type background. So he's interested in designing different ways to deliver therapeutics across the blood-brain barrier. So, the brain is completely isolated from the rest of the body by this blood-brain barrier. It's like this impermeable fence that things are not supposed to get through, which is great because it protects the brain. But at the same time, it means that it's really hard to get drugs into the brain. And so he's developed a way to actually harness sort of the body's own system using antibodies, so our immunoglobulins. An antibody is basically this Y-shaped structure here. And what he's done is he's actually modified it so that it's going to target very particular types of receptors that sit on the surface of cells, of neurons. And so some of these actually they're going to shuttle in and out really fast, okay, across the blood-brain barrier very, very quickly, in and out. And others, like this one, the CD98 heavy chain, it actually goes really, really slow. And so it gets retained in the brain for longer, which means that now if you actually kind of hook this up to an antisense oligonucleotide, for example, or some other type of therapeutic molecule, you can actually get it across the blood-brain barrier and into the cells that matter at a much higher frequency. And this work is being conducted in collaboration with Dr. Sami Barmada, also at the University of Michigan. He sits on our scientific advisory board, and it's him that deserves the credit for having brought Peter Tessier to the group, is recognizing that he's got this really, really outstanding new technology that can move things into the brain. And just as a side note, this is the same type of technology that is being used by several of the large biotech companies in the space as well. So very exciting book there. I'll also tell you about work from Iran Hornstein from the Weizmann Institute in Israel. This was something really different. So what he's done here is he's taken a cell and he's labeled it with 25 different markers, which is a lot. And then he basically used some very fancy microscopes and machine learning to try to identify if he plays with TDP-43 and moves it out of the nucleus artificially, what happens to all of these 25 other structures? Normally, as biologists, we tend to look at one or two things. He looks at all 25 at once. And so this is what he labeled. This is sort of the map that comes out of that. I don't know how to explain those super well, but just to show you that they kind of group together. But what he really showed, the really exciting part of his talk, was that he showed that when TDP-43 is caused to translocate out of the nucleus and into that cytoplasmic compartment, not only does he affect the TDP-43 granules, which would be anticipated because he's mucked around with TDP-43, but he also impacted this other structure called processing bodies. And what's interesting here is that this particular structure is not one that's been very heavily investigated in ALS, but has come up every now and again. And actually at the meeting, there were 3 different groups that highlighted that there was something going on at this particular structure that I think had been missed. And so I anticipate that next year, we're going to hear a lot more about that. This is a structure that's involved normally in degrading RNA. So very, very intriguing, and we're excited to see what comes next. Okay. Everything I showed you was new and exciting. There were some updates, but there was also new and exciting pieces. But now here's some new stuff that I don't think I've shared previously. Okay, so the first story I'm going to tell you actually coming from a young investigator, Dr. Alyssa Coyne. She's at Johns Hopkins University. She has been working for many years at trying to understand how does TDP-43 move in and out of that nuclear compartment. So that nuclear compartment, that movement in and out, is governed by something here called the nuclear pore. This is like one cartoon here, it is larger. It's basically literally that, it's a pore, and things pass through it. It's really controlled. Molecules only of a certain size can pass through there freely. After that, it needs to be assisted. And what she actually showed is that there's this molecule called CHIMP7, which seems to be impacting how the components of that pore behave. And then this leads to the dysregulation of TDP-43 so that you get that transition from that nuclear into the, from that master control center into the cytoplasm. And she can show all the deficits that go with that. What's kind of cool, what she brought here to bear is based on her previous work when she was with Daniela Zarnescu in Arizona, using this fruit fly, the lowly fruit fly that invades your banana bowl probably once or twice a year. She's used this model, which has been genetically modified to manipulate this pathway, and that she actually can show deficits that are linked to, that look exactly the way that the flies behave exactly the same way as when TDP-43 is actually mutated in this particular fly. So very, very cool, showing the impact of these molecules on locomotion in these mice, which is a really, it's a neuromuscular readout. So it's a very, I could think probably one doesn't really anticipate that flies would be useful, but they really are. It's really showing that there's an effect, and she can now use this even to screen for molecules that actually restore the expression of these molecules, CHIMP7 and CHIMP2B. Another story that got a lot of attention at the meeting was from Adrian Isaacs, also from University College London in the UK. He's a relatively new investigator to Packard. He's got a brand new mechanism that he is about to publish on, looking at lipid imbalances. And basically what he showed was that there were certain polyunsaturated fatty acids, or PUFAs, that seem to be required for to support neuronal health. And so the particular ones that he identified are ones that we can't get from our diet. Our bodies actually have to make them. And so he's actually dissected out what is the pathway for that and showed that they were protective. And there's lots of future questions now that he's generated about what are the potential interventions that one could use? Are there particular genetic risk factors, since basically our bodies have to make these? And so are some people making them better than others? For example, these are all questions that are being asked. And so just wanted to highlight here that, again, this particular, in the blue here, this is a figure from their recent paper. They use that fruit fly model, it's also called Drosophila. They used fruit fly models of C9 or 72, as well as using iPS-derived neurons for patients having the C9 ALS, C9 expansion. So very exciting that when you manipulate this particular polyunsaturated fatty acid, that you can make things better for the neurons. Next is a story from Rita Sattler. She presented a brand new story exploring a novel protein called Sparkle1. I just love the name of this one. Sparkle1 is not even expressed in the neurons. It's expressed in the cells that are around the neurons, these astrocytes, also called glial cells. And basically its job is to stabilize how the neurons connect to each other. So it stabilizes the communication of one neuron to another. And so she was leveraging other previous work that she's published showing that Sparkle1 was dysregulated in C9 or 72 patient brains. And I highlight this story because it emphasizes the idea of non-cell autonomous loss of the motor neurons. So the idea, this concept means that the cell types beyond just the motor neuron are important for us, for understanding the biology. And if we need to not only be thinking about supporting the neuron itself and studying the biology within the neurons, but also studying the biology of the cells that surround the neurons and help support them. So this was Dr. Rita Sattler from the Bear in the Phoenix. And the last little tidbit I have for you is one that I just added quickly this morning because I was like, oh yeah, this was super cool, super controversial, I'll be very honest, from Phil Wong, also from Johns Hopkins University, in which he talked about the idea that he was able to detect TDP43 pathology. So I've used this figure many times, and so all of these images, except this one, that's the control, all of these images refer to TDP pathology. And that he identified it in SOD1 cases. And so remember this piece of the pie, I told you 97% of ALS cases have TDP43 pathology. That 3% that doesn't are the SOD1 cases and the FUS cases. So patients that have FUS or SOD1 mutations. And he has actually said that now he can detect it in the SOD1 cases. And this was very, very surprising to many in the room. But there was a young researcher also in the room from the University of Aberdeen in Scotland who also had just published the exact same week as this meeting, that she also was detecting, using a very different method, detecting features of TDP43 in SOD1 cases that look similar to cases that were carrying TDP43 mutations. So I think it's early days. We've got lots of work to do to understand whether or not TDP43 really does have something to do with the SOD1 genetic context. The other, a little tidbit of the puzzle, which is related, kind of, not really, but kind of exciting, is that Dr. Wong also showed that there is TDP43 pathology in Alzheimer's disease. About 30% of individuals with Alzheimer's also are showing dysregulation or mislocalization of TDP43. This has been reported by several other groups now as well. We're still trying to understand, but it's interesting that we've got different types of neurodegenerative diseases in which there seems to be some sort of common dysregulation of what I think TDP43 is a master regular. I call it Goldilocks, actually. TDP43 has to be expressed in just the right amount, in just the right time, in just the right place, so that it can do all of its many functions. So those are the stories that I wanted to highlight to you. The takeaway messages are the same as what I highlighted in previous iteration of this talk, is that in order to get therapies and biomarkers to clinic, you have to understand the biology of the disease. And so we need to do this fundamental research that Packard supports. And as I hope you'll notice, it's collaborative. There are lots of different collaborations that are happening amongst many of the groups that I mentioned to you. I didn't highlight all of them, but almost all of these investigators are sort of cross-connected to each other through various projects. It's a global effort, people around the world. And so these investments in preclinical lab-based studies are really, really important. And that's how we're going to meet our mission collectively, is to find these therapies and biomarkers for patients. So I want to just land here with reiterating our message of hope. There is, I think, a significant amount of hope in the field right now. We've had some fantastic research that is happening. We've had some successes that have happened, of course, with the approval of tofersen or Qalsody as well. And we've also had some disappointments, and we understand that. But we are learning from them, and we have quite a bit of hope going forward. If you'd like more information, please do reach out to the packardcenter.org. With that, I'd be happy to take any questions.
That's terrific. Thank you so much, Dr. Vande Velde. Wow, I love how you give a snippet of so many of the important talks that happened at this meeting. This was just really awesome and informative. I just want to remind anyone, if you have a question, just pop it into the Q&A function at the bottom of your screen, and we'll be sure to ask that. Just a couple of questions have come in so far. I think earlier, sort of in some of your earlier slides, you might have talked about a diagnostic test, and someone's asking how specific is that diagnostic test that you mentioned?
Maybe that was referencing the HD-GFL2. So perhaps, so it's, as of right now, I think it's not as specific as it needs to be. I think would be the honest answer. It's really, and I think that it's not really being thought about so much as a diagnostic as much as a prognostic marker. So the idea of being able to identify when a patient that is a carrier, a gene carrier, might convert from a pre-symptomatic or an asymptomatic state to a symptomatic state, and catching that before there's a rise in the neurofilament levels, which would be indicative of neuronal damage. So I think, in all honesty, I think the test is not yet as specific as it needs to be. And I think that's in part because we understand now that TP43 pathology maybe is occurring in a much broader cohort of patients than we thought, as I mentioned about the Alzheimer's cases, for example.
Great, thank you. That's really helpful. There was a question that was submitted during the registration process ahead of time, and you might have talked about this on one of your slides. Someone asks if there's any new research regarding TBK1, if you're able to comment on that.
So TBK1, I think, was covered maybe in one of the talks this year. So TBK1, gosh, I have to check my notes. I have like 30 pages of notes from this meeting. TBK1 is an interesting gene in that it's, I remember now, it interacts with optineurin 1, has to do with the autophagy pathway. I'd have to say, if it's possible that I can come back to that question, I'd be happy to answer that by email, because I don't want to make a mistake about the TBK1 story.
No problem. Yeah, we can easily get back to this person.
Yeah, I'd be happy to do that.
Interesting question just about the Packard meeting, asking if you can explain how the speakers and the topics for the symposium are chosen.
Sure. So a couple of different factors go in. So one of the requirements of receiving funding from the Packard Center is giving an annual update on the research that's been funded. And so anybody that is a Packard funded investigator is actually obligated to speak at the annual meeting and give an update, and they are actually being evaluated by the scientific advisory board in the room. So that's a large portion of the program. We also have what I would say, I call them my prospect pile, in which perhaps, either I've seen them at a meeting or members of our scientific advisory board have seen some work and during their travels, and they submit names to recommend to speak. And so we have some portion of the program are sort of what I call the brand new faces, new project, new PIs. So I personally have been focusing on trying to bring in some of the young investigators. There's been a lot of fantastic new talent that's entered the field. And so it's an opportunity that works both ways for Packard to see what is the new and exciting stuff and get it on the ground floor, as well as for them to get the exposure to the larger Packard network of investigators. I also actively talk with a lot of people of trying to look at what is the stories, the newer stories that are out there. And so I chat with people here and there at different meetings, as do other members of the Scientific Advisory Board. And so that's how we kind of pull the program together. We also, in particular, in 2025, one of the things that I didn't highlight so much was that we actually had a extra afternoon of talks that was sponsored through Answer ALS, with the idea of focusing on computational biology, big data, machine learning type talks. And so there was a significant amount of the program that went towards that this year.
Thank you so much. And I love these next two questions because I think it's related to this topic around hope tied to ALS. One person asks, if you were going to summarize in very brief, concise terms, what would be the most significant discovery that you feel brings the most hope to patients with ALS at this point in time?
Wow, what a great question. Big question too, not an easy one to, it's like trying to ask me, I think we've had this before, in which I said it, I think my response was, it's like trying to ask somebody who's your favorite child, right?
Yeah.
Because like it's a hard one to answer, because I think we've made such incredible strides in understanding TDP-43 biology and this concept of cryptic splicing, and the idea of how we're going to exploit that is important. One of the other things that I think actually stems from something that I think maybe some people would not say is a happy story, which is, of course, the C9orf72 trials of Biogen and Wave. They failed to meet their primary and secondary endpoints, which was a huge disappointment to the field. These are the antisense oligonucleotides that were targeting the C9orf72. But I'd like to see the silver lining in that, which is probably going to sound terrible to some, but that it actually told us something. It was a real live experiment. I know I'm not supposed to use that word when it comes to talking about, a clinical trial, but it was a really clear experiment that told us that maybe we don't have all the story. And so what I've seen now in the last year is a last year and a half, a significant, almost a rejuvenation, like a real energy focused on trying to understand the C9orf72, because the original premise of why those antisense oligonucleotides move forward to clinic doesn't look like it was right. And so I would argue that a failed trial, so when it doesn't meet its primary and secondary endpoints, is not a failed trial. It actually told us something. A failed trial is one that was not designed properly, not executed properly, and you ended up that you got a bucket of mud at the end that you don't know what you've got. And so the question didn't get ruled out or ruled in. At least with the C9 ASOs, the trials that were done, it ruled out something. And it's caused a significant re-energy, a refocus on, okay, we got to figure this out. And so while there was, I think we're still very early, but I think I was at a different meeting recently, a C9 meeting, and I started to see, you started to see the snippets coming. It's like, you're trying to get a picture in focus, and it's just starting to come back into focus again. And I was like, okay, this is great. So I'm very excited about that, because I still think there's a lot of hope there in the C9 story, that we're going to get somewhere, and that the next trial, we're going to learn even more when it's ready. Other things
the pictures of the puzzle pieces you have too, it's great. It just sort of demonstrates that over the last 30 years.
Yes, we've made significant progress. I mean, we have, I think one of the other things too, which is maybe hard maybe for some to appreciate, the field has grown. Like, there's just so many people that are focused on trying to understand this disease. There's a lot of groups, and the next generation, you see the seeding. The Packard meetings, when it started 25 years ago, was just a couple of groups, right? And then when I was a postdoc and I would go to this meeting, it was still not a lot of groups. It was a fairly small meeting, less than 100 people. And as I started at the beginning, this meeting was over 300 people in a packed room. No, it's standing room only. We have a difficult time actually to manage the guest list, if you will, because we understand that people want to be there. They want to hear the unpublished science and advance their work and talk to their peers and establish collaborations. And we struggle really hard with trying to make sure that we try to get as many people into the room as we can. But at one point, the fire marshal is going to be like, "That's it. You can't have any more people in the room." Which is why we have a virtual component to the meeting, so people can still, Zoom in and listen from their various labs. And so we've tried really hard, for example, is that, labs, we try to make sure that it's, there are lots of labs with maybe 1 or 2 people coming from the labs versus, a handful of labs that are bringing 10 to 12 people, right? So as a way to try, again, to encourage more interactions and this amazing community of researchers that are all working towards the same thing.
Thank you for that. It's such a great comprehensive answer. And the last question we have here sort of ties to this topic too, thinking about, we've talked a lot about genes, right? And the DNA behind what might be causing ALS. And this person's mentioning that it seems a lot of the current work might be related to genetic or familial ALS. And how does that maybe tie to sporadic ALS and just, things to think about and what to get excited about in that arena too?
So 100%, I hear that and I understand that. So the one thing there's 2 things I would first emphasize. So TDP-43 is not only in familial ALS, it's in sporadic ALS. And so there's so much emphasis. Yes, there are TDP-43 mutations that cause ALS, which I didn't talk about. They're extremely rare. They happen, but they're quite rare. I think it's 4% of all patients worldwide is what we understand right now. But that mislocalization, it's 97%, maybe 99%, depending on whether Dr. Wong's data is right and Dr. Gregory. We'll find out in the years to come. So that work does have relevance to sporadic ALS. Also, just to reiterate that the C9orf72 expansion, it occurs in 11%, at least 11% of individuals with are so-called sporadic ALS. Mm-hmm. The other thing that I would also highlight is that it's not that we don't study sporadic ALS. There are many groups that are using patient-derived cells, so iPSC. So, for example, a patient can give a skin sample or a blood sample, and those cells then can be reprogrammed to be what they call induced pluripotent stem cells, or iPSCs, and then using various, I call salt and pepper techniques, you can add a little of this, add a little of that, you can make those cells turn into the motor neurons that are lost in ALS and study them that way. And so the Answer ALS platform, for example, is one of those where there's 1,000 cell lines that are available for individuals, for research groups, in which they can study whether or not this biology that they've identified, is it also common in the sporadic ALS cases. And so Dr. Alyssa Coyne's data, in which she's looked at the nuclear pore complex, she's looked in over 200 lines, many of which were sporadic lines, and she sees a very common phenotype between them. So we are studying sporadic ALS, but the only way to model it, if you will, is to actually use these iPS-derived neurons.
Thank you so much, Dr. Vande Velde. I'd love to, a couple more questions that have come in. This person says, to clarify, people who don't have familial C9 ALS may end up with the C9 expansion as a part of their progression. Is that right?
Can you say that one more time?
Sure, just trying to clarify around the genetic type. So asking people who don't have familial C9 ALS, could they end up with the C9 expansion as they progress?
So not as they progress, right? Because the expansion is either there or it's not. Right. But you can have non-familial ALS or sporadic ALS that is attributed to a C9 expansion. One of the things, one of the realizations that I've seen in different meetings this year, and it's getting talked about a lot more, is the idea that the C9 expansion is not 100% penetrant. By that meaning, it's not that it's 100% of the time that there's an expansion that that person is going to transition to an ALS or an FTD phenotype. And that is an idea that is really starting to take a foothold in the community. There's good evidence for that from studying of patient data. It's not like, other mutations, it's a 1-for-1. You have that mutation, you're going to get the ALS phenotype. C9 is not always that, is what it seems to be coming out to light right now.
Thank you. That's really helpful to clarify. Thank you.
Mm-hmm.
I don't see any other questions. And wow, this was so informative, so helpful. Wonderful to hear from you, Dr. Vande. Just the great recap of so many wonderful talks that happened at Packard. Thank you so much for being here tonight. I think we have a few final slides. I really just want to thank everyone for joining us tonight. We're grateful for each of you for your time and commitment and just for being a part of our community. Once again, a big thanks to all of our sponsors for their support. And the ALS Network brings our community together in various ways throughout the year to create help and hope for families impacted by ALS today and for those who will be diagnosed in the future. These celebratory and meaningful gatherings provide opportunities for connection and to raise awareness and funds to benefit the ALS Network's mission priorities. Please visit events.alsnetwork.org to learn how you can join us. And as you know, philanthropy fuels everything we do. We're so, so grateful to generous donors who make gifts of all sizes throughout the year. To learn more about how you can support valuable programs like this one tonight, please visit our website at give.alsnetwork.org. And watch out for the survey in your email to give us some feedback about programs like this one tonight and help us plan for future programs. Thank you so much, and we'll see you next time.
Thank you so much, Audra.
Thank you.
Good night, everyone.